Hello dear students, myself Dr. Tanushi Chatterjee from Department of Biosciences, IMS Ghaziabad University Courses Campus. Today I am here to deliver a lecture on Streptococcus. Okay, so the contents for the today's lecture will be general characteristics, morphology, clinical manifestation, classification, laboratory diagnosis and treatment related to the Streptococcus. So let's understand what basically a Streptococcus is. Basically, Streptococcus is a gram-positive bacteria which is in a spherical or ovoid shape, and they are arranged in a chain form. That is why they are known as the Streptococcus. Coccus means spherical or ovoid in shape, and they are arranged in a chain form. That is why they are known as the Streptococcus. So they can be arranged in a pair form or in a chain form. So they are pathogenic also. What is the pathogenic? That they have the virulence capacity, that they can cause infection in the human beings, in some animals, in the living beings they have the potential to cause some infection. They are some non-spore forming bacteria, but in some cases or in some conditions they also form the capsules. What are capsules? Capsules are the outer layer of the cell wall which it develops because of the deposition of some polysaccharides and all these things. The formation of capsules makes the bacteria virulent also. Basically they are catalase-negative and they are non-spore forming, non-motile bacteria. So these are the general features or the general characteristic features for the Streptococcus. Next we move on to the classification of the Streptococcus. If we talk about the classification, they are broadly classified on three types or under the three categories. First, in the requirement of the oxygen. If we talk about the requirement of oxygen, they can be aerobic or they can be anaerobic. What are aerobic species? Aerobic species are those bacterial species which grows well or proliferate well, replicate well in the presence of the oxygen. And on the other hand, what are the anaerobic species? Anaerobic species are those species which grows well in the absence of the oxygen. So basically, if we talk about the anaerobic species of Streptococcus, they are the Peptostreptococcus, and aerobic species, example for the aerobic species of the Streptococcus are the normal Streptococcus. Next category for the classification are the hemolysis. What is hemolysis? The breakdown of the hemolysin enzyme when they are cultured on a blood agar plates, they show the hemolysis. On that category, it is divided or it is classified, defined as the alpha hemolytic, beta hemolytic or the gamma hemolytic. So we will define or we will explain in elaboratory form, in a well mannered form in the next slides. Second classification, that is the third one, is the last one, is the classification for the Lancified classifications. Lancified classification defines and the carbohydrate antigen, which is also known as the C antigen of the beta hemolytic Streptococcus species. So on the classification, there are three types of classification: on the basis of the oxygen requirement, then on the hemolysis, and then on the Lancet classification. Next, what is hemolysis on blood agar? Just a few minutes back I explained you about the hemolysis. Basically, what is hemolysis? Hemolysis is the breakdown of the hemolysin enzyme when it is grown on a blood agar plates. So first type is a alpha hemolysis. Alpha hemolysis is a partial hemolysis in which it is, let me show you the diagram. First one is a beta hemolysis. In this you can see the culture plate, that is there is a clearing zones in the around where we have the cultured the bacteria. It is showing the complete hemolysis. Where it is showing the complete hemolysis, it is known as the beta Streptococcus or the beta hemolysis. Next one is the alpha hemolysis, where it is showing the partial hemolysis. The second diagram is the alpha hemolysis, where it is the partial degradation or the partial hemolysis is there. And last one is the gamma hemolysis. Gamma hemolysis is where there is no hemolysis is caused by the bacteria which are grown on the blood agar plates. So basically on these three types it is divided as the alpha hemolysis, beta hemolysis and the gamma hemolysis. Next, the third classification is based on the Lancet classification. Lancet classification is explained as the carbohydrate antigen of the cell wall. On this way, it is being classified. The classification exists into A to the V category or the groups. But in case more frequently, if we say more frequently, we use the A to D, that is a group A, group B and group D. So these are the few examples which comes under this category, that is the Streptococcus pyogenes, which comes under group A. Next one is group B, that is the Streptococcus agalacti, and the group D, which explained is the Streptococcus galactolyticus. These are the main classification for the Streptococcus. So if we say there are different strains of Streptococcus which comes under the Lancefield classification. The Lancefield classification is explained around A to V category, but the more frequently which is being used, that is the group A, group B and the group D category. Next one, the disease which are associated with the group A, group B, group D are the group A, there is Streptococcus pyogenes, which causes pharyngitis, rheumatic fever and the scarlet fever. Next one is the B category, group B, which comes or the species which is the species or the strains of Streptococcus which comes under group B are the Streptococcus agalacti. These causes neonatal sepsis or the meningitis. Group D, Streptococcus galactus and Enterococcus species, which causes endocarditis, UTIs, GI tract diseases. These are the diseases which are caused by these kind of species of Streptococcus. Next, the clinical manifestation which is associated with the Streptococcus are the toxin mediated diseases, because Streptococcus possesses some types of toxins in itself. These toxins can be streptolysin O or the streptolysin Z. So these toxins, when it is released in the cells or some organ or some part of the body, they start causing infection, and these are the virulent factors also for the Streptococcus. So this cause is the scarlet fever, necrotizing factor, which also lead to the necrosis, and necrosis is also leads to the fatal condition which causes apoptosis. What is apoptosis? Apoptosis is a programmed cell death. So gradually if a person suffers from the necrosis, it causes the apoptosis. And last one is the streptococcal toxic shock syndrome. So these are the toxin mediated diseases. What is toxin mediated diseases? When the toxins released by the Streptococcus species is released to some organ or the site where the infection is occurred, is if this toxin starts releasing and that is damaging that particular area or that particular site, then it comes or it is defined as a toxin mediated diseases. Next one is the pathogenesis and the virulence factor. What is pathogenesis or the virulence factor? Virulence factors are those factors of a bacteria, of a microorganism, which is responsible for causing the disease in an individual. So the pathogenic organism or the pathogenic part which is responsible for the causing the disease in particular individual. So what can be these virulence factors? They can be some structural components, they can be some enzymes, or they can be some hemolysins or the enterotoxins or the toxins. So if we talk about the structural components, what it can be? It can be the M proteins, it can be the lipoteichoic acid, it can be the capsule, as we are talking about the structural components. And these structural components may mostly comes under the cell wall components also. Next, what can be the virulence factors? They can be the enzymes which it is being released by the bacteria. The enzymes which associated with these diseases or have the potential to cause the infection in the individual, they can be the streptokinases, deoxyribonucleases, high aluronidase. These are the enzymes. Next one is the pyrogenic or anthrogenic entotoxin, exotoxin, which are the last in the last slide only I discussed about the toxin mediated diseases. So these toxin mediated diseases are caused by the toxins, and these in case of streptococci, what are the main toxins? Streptolysin O and streptolysin S are the toxins which is being released by the bacteria and responsible or is have the potential to cause the infection to the individual. Next, coming to the laboratory diagnosis. What are the laboratory diagnosis? If we initiate with the laboratory diagnosis, the first step have to be the sample collection. For the sample collection, we may use the sample as skin swabs, throat, sputum, urine and blood. After that, the first what we do, we go ahead with the gram staining, because it is a gram positive bacteria. So we have to explain, or we have to identify the whether the bacteria is a gram positive bacteria or it is a gram negative bacteria. So if it is a gram positive bacteria, then we can go ahead with the culture part, as we have identified or we are suspecting it as a streptococci. Then we'll go for the blood agar culture plates, culturing plant. Blood agar culture plate, if we are using the blood agar culture plate, then we also use two types of antibiotics, that is the optogen and the bacitracin. Optogen, it also may give the positive results, means it may be sensitive or it may be resistant. In case of bacitracin, it may be positive or it may be sensitive or it may be resistant. If it is, but mostly the optogen antibiotic is used in case of the alpha hemolysis, and bacitracin antibiotic is used in case of beta hemolysis. So further on this sensitive and resistant clearing zones, the zones which are being observed after using these antibiotics in the culture plate, it is further classified or further identified. It helps in the identification of the bacteria. The detailed thing will be explained in the next lecture. So next comes to the catalase test. Catalase test is performed by using the hydrogen peroxide. When a drop of hydrogen peroxide is placed, or is in the drop of hydrogen peroxide is placed with the culture, then it shows a fuzzy reaction. The reaction which occurs is it degrades into hydrogen, means water, H2O and oxygen. So basically in case of streptococci, it gives or it provides negative results. Then coming to the treatment, as it is a generalized lecture, so what basically it contained, that the treatment, how if a streptococcus infection is affecting an individual, what is the baseline treatment? That is being treated with the penicillin. Mostly it is being treated with the penicillin. But in case of few individuals who are who cannot take the penicillin or is not suited to that, in that case erythromycin can be given to the particular individual. In case of some allergic reactions, some individual may not suit with the penicillin, so in that case erythromycin can be given. So these are the baseline treatments, the basic treatments or the basic antibiotics which can be given to the particular individual for treating the streptococcus infection or the infection which is being caused by the streptococcus species. Now the preventive measures, why this infection will cause, so avoiding these kind of infections, so that a particular individual or a person should not get the infection from a streptococcus species. In that case, what measures can be taken, that it should be hygienic and the proper hygiene should be maintained. Or if you are having some kind of throat pain or something like that, you should take proper gargles with the betadine or some warm water with some adding some salt. So if we're taking these kind of measures, it may prevent the further infection or the severity of the infection. So basically, as it was a introductory lecture for the streptococcus, we just had the introduction about the streptococcus. What is a streptococcus? It is a gram-positive bacteria which is in cocci shaped and it is arranged in a chain form. Then what is the classification? Basically the morphology, it is a cocci morphology, and the classification based on the oxygen requirement, then hemolysis, and then Landsfield classification. Then what the clinical manifestations or the disease which it can cause, the different types of streptococci species, and then what are the laboratory diagnoses which can be pursued or which can be done to diagnose or to identify the person or the bacteria which or the strain which infection is caused, which type of strain is causing the particular infection, that is. And last one is the treatment. So this is all for the today, and in the next lecture we would like to go with the brief more deep study about the streptococcus and the bacterial infections. Okay, so thank you students, see you soon after this one. Thank you so much.